People talk about the focus of are the technologies ready? We can make generally the products we're trying to make. We're not in the industrial space making them at a large enough scale at a low enough cost. And our belief is without a purpose-built solution that the industry as we know it today will stall.
Hi everyone, in today's video we're going to go through an excellent interview by the CEO of Liberation Labs. Liberation Labs is building infrastructure for fermentation. So they're building the actual manufacturing capacity so a lot of these food companies can use their fermentation capacity to ferment the ingredients that they need to make their food products. Before we get into that, I just want to do a quick note for Agronomics investors. Like when this company was first founded in 2022, they were one of the initial funders. And as of that initial funding, they own about 47% of the company. Now, why is that important? This is an article from TechCrunch from about 6 years ago, so it is old, but I just want to quickly go through a couple points before we get to the interview because it really explains well my style of thinking about investing in VCs. So who's actually succeeding in making money? A VC fund needs a 3x return to achieve what is considered to be a good rate of return on their investment. So a 100 million fund needs to have a 300 million return. The graph below shows what percentage of VC firms accomplish this. As we can see, only 5% return more than 3x. The majority return less than 1x, 35% return 1 to 2x, 10% return 2 to 3x. So most are juggling somewhere between breaking even and losing money. Remember, we have inflation. I'd encourage everyone to read through this article if you've not seen it before, but essentially there's a couple metrics that we should be looking at. Venture capitalists get their money from high net worth individuals, family offices, that kind of thing. And so there's a lot of ways for them to make money. And if they're going to make money, for them, success means getting a 12% return per year. So a 10-year VC fund needs to return 3x in order to earn that 12% per year. 80% of the returns come from 20% of the startups. So let's look at just one scenario. So we have 10 startups and a fund that needs to return 3x within 10 years. So let's assume it's a $100 million fund with $10 million invested in each company over the course of its lifetime. Let's also assume that the VC jumped in on the A round, followed up on the B round, so added more money, and has a 25% ownership at the end. Let's look at the different outcomes. So outcome 1 is they all do average and exit at 50 million. Green marks the exit size, 50 million. Purple marks the payout amount of the VC with 25%. So the VC gets 13 million. Outcome for those 10 companies, VC would return 12.5 million each. That's 125 million, but we need 300 million. Okay, so let's increase the odds that they do better. So if we're getting 5 selling at 50 million, 5 selling at 100 million, that gives us a total of just under 200 million. Still not quite there. So the majority do average, and we'll throw in an overachiever. So 5 at 50 million, 4 at 100 million, 1 at 500 million. That gives us 287.5 million. So what do we need? We need a unicorn. All 9 sell for 50 million, 1 sells for $1 billion. That gives us a 250 million return. So in total, we get 362.5 million. But can we realistically expect all 10 companies to exit? Why I bring up that Agronomics has a 50% stake in Liberation Labs is to illustrate this point. When investing in Agronomics, you're going to need to figure out who are the winners, what percentage does Agronomics own, which they do excellent job at providing those details. Then you're going to need to actually follow the companies that are these companies that should succeed. So here's another article, and I'm going to leave this in the notes as well below the video, but this is just a good way of seeing things. So the VC-backed businesses, by default, they fall into three buckets. The rock stars that aspire to become a unicorn. So this is that 1 billion exit that we're talking about. So a VC fund will want to invest as much capital as possible in this company, right? Because they want to get this return so that they can meet their goal. The dark horses is where a VC will invest energy, time, and provide access to their network in hopes that they become rock stars. The walking zombies never really find someone that actually wants to buy their products on a meaningful scale. So these are kind of the three buckets that you can put these companies into. These are just some useful frameworks that I use to think about investing in VCs. So if you are interested in agronomics or do invest in them, this is one of the companies you should keep on your radar. One of the interesting things is they recently did another funding round when they started to build their latest plant in Indiana, and that was a debt financing round. And so that doesn't give away any equity. So that's important to understand as well, because that should mean that, at least right now, Agronomics still owns a sizable chunk of the company. Whether you agree Agronomics is a good investment or not, this is a really important company to watch because they are building so much of the needed fermentation capacity. It's worth noting that the heme for the Impossible Burger and the egg protein from the Every Company are both manufactured with Liberation Labs. And I'm sure there'll be a lot more large companies signing on in sometime in the near future, especially once they are able to launch this factory later in 2024 and then ramp up that production.
Our reason for getting together is to hear from Mark Warner at Liberation Labs. And before we get into a little bit of a fireside chat format, Mark is going to present a few of the key details of the Liberation Labs investment. Mark, to you.
Great. Thank you, Matt. Appreciate the opportunity. As Matt said, just going to give kind of a brief summary of what it is we're doing, both high level as a company and then specifically with our first facility in the US. So we'll go ahead and dive right in. So with the founding of Liberation Labs, our real goal is to become the global fabrication partner for industrial biotech. And I want to focus on the fact that we are focused at industrial biotech, novel foods, materials, other products, basically everything but APIs. So just realize when I'm making my comments about the market and what we're targeting, it's targeted at the industrial side, not at the pharmaceutical side. So with that, we'll go ahead and jump in. One thing that I want to talk about is the global nature. As I mentioned, we're starting in the US, but we're looking to get a global network because we believe it's important for a lot of these products to get wider scale acceptance than they are today, to be able to use a network that when a CPG launches a product in one geography, to be able to move to the next geography. The other things, and I'll kind of step back and say, what's out there for the network we have today? As a consultant, I consulted for about 8 years, worked for over 100 companies in the novel materials, novel proteins, cellular meat space. And as I was helping them find contract manufacturers, kept running up against a few obstacles. First, the existing network out there is about 40 to 50 years old. It was built predominantly for pharmaceuticals, chemicals, and biofuels. Generally, the fermentation is a closer fit to what's needed. The downstream is most likely non-existent or not a good fit, and very often has to be built. And where these facilities were located are basically where it made sense in the 1940s, 50s, and 60s to put generally an antibiotics facility, not where you'd put a novel protein, novel materials facility. What this all rose down to is the majority of them are not fit for what we're trying to use them for today, and that they don't have viable system economics. A lot of our premise is People talk about the focus of are the technologies ready? We can make generally the products we're trying to make. We're not in the industrial space making them at a large enough scale at a low enough cost. And our belief is without a purpose-built solution that the industry as we know it today will stall. So let me talk about what our approach is. This is more than a figure. This is actually out of our 3D engineering CAD model of the facility we're building currently in Richmond, Indiana. It's a 600,000 liter facility. It's what we're calling our launch facility. It is commercial, it is profitable, but it's on the lower end of what you would consider commercial scale for industrial biotech. We plan later in this geography to expand up to about 4 million liters. We also expand to go into additional geographies around the world, which is a little outside of what I'm going to talk about today. But let me drill in a little deeper. When we decided what it was we're going to build, we weren't hampered with a legacy facility built for something else. We stepped back and said, what are the most common organisms used today? And what are the most common downstream processes? And how do we build a facility that we believe can host the biggest swath of these processes? So we targeted really the novel protein space. And I like to say things that ferment and are recovered like a protein. It doesn't have to be just a protein, but in the center, the two tall, large green vessels, those are our fermenters. This facility will have 4 fermenters, production fermenters of 150,000 liters. These are high oxygen transfer, 150 oxygen transfer at 28 degrees C. We'll run most processes. Our 2 base organisms that we kind of designed the bulk of the facility around were a methanol-fed Pichia pastoris, one of the most common grass-approved food organisms today, and E. coli, very common both in industrial biotech and pharmaceuticals. It doesn't mean that we can't run other yeasts and filamentous fungi and other things. We're confident we can. We believe they fall within the parameters of those 2 bounds. The downstream process is a high-efficiency, multi-stage ceramic microfiltration, a high-efficiency, multi-stage spiral wound ultrafiltration, evaporation, and spray drying, all on the same site. And for those of us who have been to a lot of these CMOs, a lot of people who haven't don't realize this doesn't often exist on one site very often. So to kind of talk about what we think makes our facility different, it's purpose-built. And I get asked a lot, what do you mean by purpose-built? We believe this is the first industrial biotech facility in the world built for a methanol-fed Pichia pastoris. Doesn't mean other older facilities that have done their best to retrofit can't run the organism, but they were never designed for it, and they can't do it as cost effectively as we believe we can. Our knowledge comes from what we built working both for companies in the space and the existing CMO network to understand what it is we think needs to be built. We believe this gives us significant agility and flexibility, very favorable economics. As I mentioned previously, a complete solution on the same site. I can tell you as somebody who's built and managed large commercial fermentation facilities making food products, that doing it all on one site and doing it very quickly is very important for food products to keep things not just the ultimate food quality and contamination, but even things like off notes and flavors. All of these components, we also believe, lead to a much lower environmental footprint than the existing facilities that are out there today. So, I've talked about what we're building. Let me compare it with what's out there today. So this chart, when I was consulting, I tracked about 65 facilities worldwide that did contract fermentation of industrial biotech. These are the top 25, I think it's 25 facilities, represent like 70% of the capacity. Size of the bubble is the fermentation capacity. Bottom is the year it was built, roughly. Left side is our rating of how ready it is to make something like a food protein. Higher up you are on the list, it means they can ferment and have the capacity to do the downstream processing. Lower on the list, generally they have the fermentation, but not the downstream or the experience in food proteins. There's a couple takeaways here. I often tell people, you know, this comment of the existing network averages 40 to 50 years old, but until you see the dots on a chart, most of these facilities were built in the 1940s to 1960s, as I said, to make antibiotics. Some more recent ones for chemicals and amino acids, but the bulk of the network out there today is at the end of its useful life. It's also not as efficient as a purpose-built facility. So this is the gap we're looking to bring in the upper right. Both, you'll see the initial smaller circle of the 600,000 liter facility and the full 4 million liter facility. We think it's coming to the right geography in the US. That's where, you know, when you look at the product offtake, most of these novel proteins can only be sold in the US. So the fact that they're having to be manufactured generally outside the US today is a cost and a factor that's limited their adoption significantly. So I'll kind of wrap up with the last slide. This is a little deeper dive, and I'm not going to go into all the details, but it highlights for those with more of an engineering manufacturing focus. When we say fit for purpose makes a difference, what do we mean? The style of fermenter, its high oxygen transfer agitated, how that compares with what's out there today. This ability to feed methanol. We can feed methanol to 100% of our fermentation capacity. It was not a major capital cost item for us to add to a new facility, but it is very difficult for older facilities to add it, to the point when I was consulting, a lot of these older CMOs would try and pressure companies to take methanol out of their formulation, which, you know, think about that. We have older CMOs trying to get processes adapted to fit their existing network, as opposed to building what we need. Things like high volume, high turnaround, sterilization, quick turnaround times. Kind of the last two I want to highlight, the amount of product we can make out of the equivalent fermentation capacity is generally, we believe, about twice what most of these facilities that are downstream limited can make, and how quickly we can do it. We're designed for a high throughput. We can run our facility, I should have mentioned this, the 600,000 liters, we can run in 250% trains. I can run E. coli in one half. I can run methanol fed picky in the other half, or I could run the entire facility on one train. The only thing we do campaign is the dryer, but we're set up through fermentation. Once it drops in fermentation, microfiltration, ultrafiltration to the feed to the dryer within 2 days, much quicker than most facilities out there today. So with that, would welcome Matt's questions. And please, as he said, if you have any questions about what we're building, the industry in general, would love to field any of those questions.
So, Mark, we have a couple of questions popping up in Q&A, but before we get to them, I wanted to ask you to talk about how you got to this point. And you'll recall that the first moment I caught up with you in the lobby at SynBioBeta, the first question I had for you was, why Richmond? So can you talk a little bit about how you ended up there?
Sure. You know, how did we end up here? You know, part of it was my co-founder, Eitan Benheim, and I, our frustration of, he was representing an older CMO, myself, that there wasn't capacity out there. We did, when we founded the company, we did look worldwide, chose the US pretty quickly in that we thought it's where the biggest gap of capacity is, and it's where most of the products are. So given that, we looked at 10 sites in our site search. We looked at both co-location, you know, co-locating next to a corn wet mill, Tate and Lyle, Cargill, ADM. We also looked at what we call merchant facilities, generally heavy industrial parks. Our search really came down to economics. The three things that make up 75% of the cost of an industrial biotech facility, different than pharma, is sugar, power, and labor. Each of those is in the range of 25%, and they come up to about 75% of the direct manufacturing cost. So we needed a place that had all three of those. When we looked at it, Richmond out of the 10 was the clear choice for a couple of reasons. It's in the industrial Midwest. They understand manufacturing. What was unique about the Richmond location, the power supply, the water, the natural gas, and the electricity, not just for our launch facility, but for the full commercial facility, all exists at the site. The second thing is I had been asked to give a presentation to the state of Indiana, similarly why we picked it. And I'll give you an example. From the time we identified the site to the time we had a buy-sell agreement for the property and a binding incentive package with the Wayne County and the state of Indiana, took us 90 days. I've had that take 2 years in many states. So they continue to impress us. We believe it's been a good match, and it's, we broke ground. We purchased the facility basically January this year. I think we completed the transaction about April, and we broke ground, within 6 months of identifying the site. So
one of the things that you said that I thought really jumped out, because obviously the current market is a really interesting set of, interesting being a kind word, the set of conditions, lots of headwinds. So in lots of ways, Mark, obviously your news, your development and your investment is exceptional because the market has not been all that friendly to biotech the last 18 months. So this is all great news, 600,000 liters of sorely needed capacity for the industry. And 100% of the companies that we've surveyed over the last year have said one of their real critical pain points is being able to find the right staff. So you've pointed out that you're within the catchment area of multiple major metros that help you solve for that. Can you talk about that a little bit?
Sure. And that was I should say, we got down to, on our list of 10, we got down to 3, well, it was originally a list of 9 that got down to 3. And the one thing that concerned us was most of the sites were pretty rural. They were, 2,000 person town, maybe an hour and a half from 20,000 or bigger town. That's when we identified Richmond. And what we really liked about Richmond, I've had the ability around the world to build and staff and start up these facilities. And just like you need sugar, power, and labor, you need qualified technical people, you need qualified technician level maintenance and things. The more rural you get, it skews towards the hourly technician type. The more urban you get, it skews more towards the professional, but it also gets a higher cost of living. What we liked specifically about Richmond is 35,000 person town, not a bad sized town, but within an hour and a half or less of 3 metropolitan areas, Indianapolis, Cincinnati, and Dayton. So we've been very happy in the mix of size, have engaged with the local trade schools there, Ivy Tech. It's a network there of, I don't know, I think it's more than just a community college, but have really embraced us in helping get that skill set. And I'll put myself in that category. Very few of us went into this saying, I'm going to be a bioprocess or biochemical engineer. I started in the chemical and engineering industries for about 20 years and moved into industrial biotech about 15 years ago. It's really only been in about the last 10 years, we've gotten really a wide berth of good bioprocessing programs. And kind of the last, I always have to give this word, we need to educate people all across the spectrum from high school degree to PhDs and from coast to coast without forgetting the middle, because you're generally going to have this manufacturing in the Midwest. Pharma can live at both coastlines. These types of facilities, just the economics aren't going to work. So the type of people we're going to hire technically are more likely to have a degree in chemical or mechanical engineering from a Purdue, a Mizzou, Illinois State, those kinds of things.
There's a reason that they're called the Boilermakers, right? There's something to that. A couple of process questions here. So I actually want to take that set of questions and ask you to slant a response, if you will. And that is, so you've mentioned the 600,000 liters. At what stage do you think ideally a prospective client comes to you? In other words, are you also going to have process development teams that can go alongside when they're at optimization stages, or do you expect them to sort of know those answers, or will you be able to work at 50 liters?
Great question. We are manufacturing. We expect people to have their tech transfer done. Doesn't mean we don't think it's critical and one of the major obstacles. One thing I've learned over the years is there's groups that are very good at scale up. There's groups that are very good at manufacturing. There's very few that do both well. So we've targeted large scale manufacturing. We've set up some alliances with groups that can help our partners do the scale up. Most of who we're talking to are operating at fairly large scale. I guess what I'd like to say is, let's talk about what we look for in a customer. So what we're looking for is somebody who's operated at least, 10, 20, 30,000 liter scale, probably for 3 to 6 months, both to get the process data they need to be comfortable scaling to 150,000 liters, that we're going to understand the economics and not just the fermentation, but the downstream, but also generally for them to confirm they have enough market offtake. Because again, for manufacturing, we're not looking to do a handful of runs. We're really mimicking more what you see in the mature pharma model of long-term multi-year contracts. When a new drug comes out, most of the companies are not manufacturing it themselves. They're going to a well-established network of contract manufacturers that were built to run those kinds of processes. That doesn't exist generally in industrial biotech now. So because of that, we're really looking for people that have that track record of running at fairly large scale.
A couple of questions too. I think I want to jump on Stephen Hubbard's question. Thank you, Stephen, for the question. He's asking about what are some thoughts that you might have about what some of the equipment companies that could do to support R&D and process innovation?
I've generally had pretty good luck with the equipment vendors. I think the biggest struggle usually at these types of facilities is there's very few companies, if any, that really you can buy a packaged 150,000 liter fermentation system. You can do that at 2,000, 10,000, but it's very few that will do a packaged system. So we've designed ours from the bolts up. It's something our team has done at other sites. It's very common in industrial biotech, but that becomes a limiting factor. So the fermentation is generally got to be designed from the bottom up. The downstream is usually packaged equipment. Frankly, doesn't look that, now for proteins, doesn't look that much different than what you'll find in a modern milk process facility. Ceramic microfiltration, some people may question, I left out centrifuges. We certainly can put in centrifuges. They were in our original design. For most of these processes, a multi-stage ceramic microfiltration has better recovery yields than centrifuges, so that's the route we went. We've got some potential clients that are asking us to put in centrifuges, and we can do it. But it's realizing that on the downstream that you need to work with these vendors, that you're probably not going to design from the ground up, and talk to them, listen to them, find out it may not be on the shelf, but you don't want to start a whole new design. It's usually going to be some adaptation of something they have.
We have a great question from one of our colleagues in Western Europe that I think is as much about perception as anything else, asking about the perceived high costs of labor and energy. And in my experience, costs of both of those inputs in the Midwest are much, much lower. Can you talk about sort of your take on those?
Correct. And this, since I think the title was U.S. manufacturing, one of the things I've made clear as I've talked at government events and stuff is, as we look worldwide, and I've built these facilities in Brazil, I've looked in Australia, I've worked on projects in Europe, U.S. has no cost disadvantage to the rest of the world. It just simply doesn't. I mean, most basic manufacturing, you always look at China and say, okay, China can do it much cheaper, to some degree India, and that's often true in pharma too. And I'd like to point out the difference, not that one of the differences is when you look at the staffing, when you look at the labor cost contribution in pharmaceuticals, it's much higher. An equivalent facility, I've done work to look at retrofitting, taking older pharma sites and putting them into non-pharma use, they often have 3, 4, 5 times the head count that a food facility does. Now, certainly appropriate. We all, when we get an injectable drug, want it to be to a certain standard. I'm not questioning that standard, but it's very different to the food industry. And this is one where, especially, Europe is where a lot of this capacity exists today in older facilities. It's challenged right now with power, but yeah, on those 3 components, the US Midwest, certainly on sugar, corn dextrose, some of the cheapest in the world, labor's competitive, power is actually pretty favorable to most of the world at this point.
Yeah, people who haven't worked on the drug side of the industry may not realize how much more stringent the regulatory regime is over it. As an example, a sample of every batch has to be saved forever to be tested long-term for quality. So the drug side just has a much higher set of regulations that are very expensive to deal with.
If I could add one more, because I was asked to do a benchmark study once, and I had a buddy who worked at Genentech and had worked at, both sides of it. And, when we're building a fermenter, we have to make sure the welding rod comes from a quality supply. That's it. Absolutely. Every piece of steel and welding rod has to be individually certified and tracked in the pharma space. Because again, ingesting something as food versus getting it intravenously, the tolerances are just different. But yeah, again, absolute night and day in what those standards are.
Yeah. Mark, interesting question about heat. A question about the heat being generated by, methanol-induced systems is the question. Did you have to do anything extraordinary in building in heat exchangers to the process to keep it cool?
There's a lot of focus on it. We designed the fermenters to run, spend a lot of time on the cooling system. So there's a couple issues with cooling, and I apologize to get geeky here, but I've spent more time on fermenter design than many. If you look at that oxygen transfer, in many historic processes, we'll say 150 oxygen transfer, but if you look at it over the batch, it'll peak at 150, but many strains come down to 100 to 120. As we've started to do more optimization, which everyone wants to do to shorten the fermentation, we're getting better about holding that 150 oxygen transfer the entire batch. You'll find a lot of fermenters do okay because they can use all the cooling in the fermenter for only half the volume at the beginning, but we had to design it to run the 150 over the entire batch. Methanol does give a little more heat, but not as much as like a methane. So it is designed into our fermentation calcs, and it's a little bit of a challenge. But again, designing those differences in up front aren't as onerous as trying to retrofit for them.
Conversion's worse. Yeah. There's an interesting question about a lot of what we experienced the last 2 years with equipment delays in building facilities. And I think added pressure for those that are not in the US, we saw simultaneous pressure on capacity shortages while also big ticket equipment was delayed with the reshoring of the supply chain that was taking place. So in particular, things like transformers and power equipment was delayed during that stretch of time. This question from one of our attendees is really as much about process equipment. Mark, do you have any sense of, it seems like we've tackled most of those equipment delays?
Yeah, I'm knocking on wood as I give this answer. So if you had asked me 3 years ago how long it would take to build Richmond, I would have told you 18 months pre-COVID. We're at 24 now. Fermenters alone, I could have gotten 4 150,000 liter fermenters fabricated in about 6 months. The fermenter vessel only pre-COVID, they're 12 months now. You are correct. And, the problem has shifted over time. A year ago, it was chips, and chips go into valves and skids. So anything with a chip in it was delayed. You were correct that when we started this process and started ordering our long lead equipment at the beginning of the year, transformers and electrical equipment were some of the longest. And I'll give you an example. We ordered traditional stuff like air compressors, boilers and stuff. And because of COVID, I'm sure everyone's got stories of, I ordered something that was supposed to be 3 months. It took 7 or 8, and I had to call the vendor and yell at them monthly for that long. Because of that, vendors don't want to commit. We joke, those of us in the engineering construction industry, we used to have like 20-week equipment, 40-week equipment, and greater than a year. we joke there is no 20-week equipment anymore now. At least they won't quote you that. We ordered equipment that had a 45-week delivery that about 3 weeks in, they notified us was going to be delivered at 20 weeks, which is good and bad. So I think things are coming back. Generally, the belief is people are putting in numbers they consider conservative. We've all just been burned enough the last couple of years that until the equipment shows up, especially if it's coming from overseas, you just don't know.
I wonder if you have similar experience with this as we did, Mark. We worked on a pilot facility during COVID and found that the tanks at 20,000 liters roughly doubled in cost during that stretch of time. So much so that the price of steel would no longer be quoted for future delivery unless you had a very specific provider with a very specific provision. So we had real trouble with stainless steel.
When we finalized our fermenter design, so stainless steel, at least at the point we ordered the fermenters 3 or 4 months ago, was unfortunately near historic highs. I don't think it was quite, it had gotten near twice, it had come down a little. So that certainly was a tough thing. It's recovered some, but it just brought this whole uncertainty that was never there. Pre-COVID, generally you could get a quote that was good for a month. And as long as, even if there was a change, you'd say, okay, we'll honor the quote, but we have to adjust, this little bit either way. But, again, we think it's better now, but yeah, stainless supply has been a challenge.
One really interesting question here is, how did the community of Richmond respond to your announcement?
Great. From the beginning that we engaged with them, the community, again, they understand manufacturing. We were a good fit for them. Like many communities, I joke, we got the incentive package in place, and I was actually asked to brief some of the community officials. A common question we get is, it's great, we love what you guys are doing. Could you tell us exactly what the products are you make? Because in the end, we're a B2B ingredient manufacturer. We'd make the heme that goes in an Impossible Burger, a Clara egg protein that goes in something else. We don't make, in the end, a product ourselves generally. We make a key ingredient that goes into something. So that's really been, some of the questions, but, there's a long history in Indiana. I'll be honest, and they've admitted this, it's that, what do they call it? Indiana modest. They don't want to tout their accomplishments. As you're probably aware, in the pharma space, they have a decades-long history. You've got Corteva in the ag biotech, and Elanco is based there. You've got Eli Lilly now at the Evonik facility. Decades-long history there in biotech, but more at the large-scale pharma, not as much until more recently in the industrial biotech.
Yeah, I think that's the original story of Genencor, right? They were looking around and Lilly had hundreds of thousands of liters laying around that they could borrow on the short term. So pretty amazing capacity. There's a really interesting question about a Nimble report or meeting output from 2021 saying that the stated view from a Nimble meeting was that biomanufacturing needs to be decentralized. Now, I would argue picking the Midwest is exactly to the point that you made earlier, that we see pharma sort of being bi-coastal and synthetic biology maybe moving to the middle. Do you have a take on whether this fits the decentralized thesis?
It depends on the product you're making and the price point. The lower the price point, which, we're talking, the food products that people hope to make, the key ingredients, at least in the, single digit per kilogram, or maybe in the teens. Some of the more niche products, maybe specialty enzymes, human milk oligosaccharides, maybe those are 30 up to 75 a kilogram. Those generally have to be in a place like the Midwest to hit the lower price points. As you get more niche and the high, if it's specialty products, and that's generally what you've found, those products I just named generally have to be made at hundreds of thousands of liters to hit those price points. Those have to be more centralized. If it's a product you can make economically at 10, 20,000 liters, it can be more decentralized. But the large volumes, and I think even today, one thing that I did this when I was at Solazyme, 10 years ago, it's a good benchmark now. If you look at something like a lysine, it's made at very large scale for a few bucks a kilogram, but it's done in very large facilities with very good performance. And I can tell you, having built a facility almost 4 million liters in Brazil 10 years ago, the economies of scale are real. If you have that need to make a product and you have the offtake at very large scale, the economies are very good. But I can also tell you, if you build that big a facility and don't need all the offtake, then of course the economics aren't what you hoped.
Yeah. So for those watching, the pressure on biomanufacturing on all parts of the industry, cell and gene therapy is the opposite. It's getting smaller and decentralized, but it's more expensive product. So that I think it directly correlates to what you're saying, Mark. I think we also know from experience that anything that's going to go into a consumer product that's a few bucks needs to be enormous volume in order to get there. Really, another great question from Dr. Humbird about process innovations like continuous. Are there particular process innovations you're excited about, and is continuous one of them?
Continuous fermentation. Facility I built in Brazil 10 years ago did a form of continuous fermentation. Continuous fermentation isn't necessarily new. It's been done in the pharmaceutical side. Now, nothing is ever forever, but, continuous is taking a 2 or 3 day fermentation and maybe running it for 20, 30 days. There's certainly a benefit to it. It usually all comes around sterility. The longer you can run your fermentation sterile, the more likely you are to be able to run continuous fermentation. Our base design is for fed batch fermentation and draw and fill. And for those who aren't deep in the fermentation space, we actually, if you look at our newsletter, we published a 10-page paper kind of digging into this. Almost all large-scale industrial processes today are at least fed batch fermentation. Very few are done at batch. People want to make references back to batch, but very few large-scale commercial fermentations are done batch. They're done fed batch, meaning I put the inoculum in, I run the fermentation till the fermenter's full, and instead of draining it and starting over, I may take half of it out and keep feeding. The ability to do that depends some on the strain. Is it, now if it's a biomass protein where you're just growing the biomass and using it, much more common for continuous fermentation. If it's something like a secreted, I'll use the methanol fed picky example, you grow up the biomass, you feed methanol to trigger it to do the secretion mode, it gets much more complicated to do it continuous. So we believe with our sterile profile and the way we're set up, we'll be set up as well to do continuous fermentation as anyone if, at the point the technologies are there.
There are a couple questions here that are awfully commercial in nature and probably premature, Mark. So for instance, I'm sure it'll be a little while before you approach the timeline at commissioning where you start to allow that capacity to be hired out. But what does that timeline look like for availability? Sounds like a few people want to get in touch.
Sure. And please reach out. We are talking to clients now. The contract manufacturing, so this facility will be up and operational end of next year. Initial operations Q4 next year, shakedown, make an initial product. Q1 2025 is when we'll really start getting into more real production. We're talking to people now, because even in my consulting practice, I probably was involved in about 20 different entities contracting with CMOs. Usually that's about a 12 to 18 month window out. We're at about 18 months right now. So we're certainly talking to multiple entities about that capacity. We hope to have the bulk of it under contract by the end of this year. So yeah, certainly would be happy for those. You can go to our website or drop me an email. Would be happy to talk to you about the capacity. The one note I would say is we are large scale, longer term capacity. So we're not looking to do a handful of runs. We're talking to people about 300,000 liter blocks. So we're looking for people that need fermentation in the range of 300,000 to 600,000 liters, which is a pretty large scale fermentation. But the other thing I'd say is we are building the whole facility, just to be clear, because I get that question a lot. we're funding the capital from sugar all the way to the spray drying on site.
Now, Mark, you mentioned too that you'll have capacity to expand at Richmond up to, I think you said 4 million liters.
Correct.
And you mentioned you'd like to do this a handful of times around the globe for the Liberation Labs forward plan. So somebody's asking a really interesting question. Is there a U.S. advantage in either in bioreactor or DSP quality in your sort of view of the world?
Is my, not really. I mean, it's all, wherever you fabricate in the world, and I'll use the example of Brazil. Brazil is the first world country in manufacturing, but it's where are you at in that part of the country? We were out in a very rural province that did sugar. We had to bring special welder. They'd never done sterile piping and stuff. So you can run into that in the US. So, as happens when you build these facilities, it's, not all the equipment. Fermenters are certainly being fabricated here in the US. A lot of the downstream equipment coming from the dairy industry, a lot of that comes out of Europe. So it is currently kind of a mix of things from around the world currently.
A couple of questions about extracellular proteins. Mark, are you going to have broad capabilities or do you expect to focus?
Well, I mean, our base design is for extracellular secreted protein. So that is the base design of the facility. We're certainly talking to multiple entities that have an intercellular. We have the ability to add homogenization or other cell disruption. It's not in our core design. There's only certain things we're really saying I won't say won't discuss. You never say that, but we're less likely to work out. And that's a solvent-based downstream. While we use methanol on the front end, solvent extraction is really more of a small molecule API space. So we're staying away from that. The main alternatives that we've looked at are homogenization or other cell disruption or addition of centrifuges. Those are certainly something we're willing to discuss. We've talked to some about adding a chromatography step at the back end. We're open to it. We question some of the economics. For those who have done, chromatography, it can, I've had chromatography resin in the pharma space that's $200 a kilogram for the resin. I've seen it in $2 a kilogram in the industrial space. I've also seen it not work. So our question on chromatography is more needing to be convinced the economics work, but those are certainly options we could look at.
Thanks, Mark. I think there's a terminology question here. So I want to actually take the camera out to, let's say, 30,000 feet. I would argue that this is sort of the third era for synthetic biology in terms of its nomenclature. The first era was the Genencore period. The second era was the biofuels craze, I would argue. And this third era is the arrival, the onset of a whole bunch of new consumer products that we've seen in the last handful of years. So Abad asks, what about precision fermentation? And I would partly argue that's just a terminology question.
I've been in debates on that. Is this the third, fourth, or fifth? I'm going to stay out of which biotech revolution it is. And what was the second part of the question?
He says, what about precision fermentation?
Yeah. That was a term, I believe, at least it came on my radar in Good Food Institute's report. And I think it's a good one. It doesn't mean, I have people ask me, does that mean every other one is unprecise? No, but they're different. Precision fermentation, what I used to tell when I consulted with people is, listen, precision fermentation is great, but whatever you're making better have a special purpose other than just nutritional value. As I used to say, it better walk, talk, and speak Latin, because, if you're making a biomass protein, you can make those at a couple bucks a kilogram in many cases, and that makes sense. That's great. Nothing wrong with that. We've chose to focus on the industrial biotech, enzymes, food proteins, HMOs, materials applications, biopesticides, higher value specialty ingredient space. I think precision fermentation is as good as any, but I will acknowledge it's not I hate to say not the most precise term.
The Department of Defense has had trouble with this terminology for years, right? The engineering biology phase that they went through, the terminology changes as we keep seeing. Dr. Hubbard's now trying to get us in trouble, Mark. So do you have an opinion about the the techno-economic analysis that Humbird published on cultivated meats?
You know, I was actually asked I was a peer reviewer of that report. So, you know, I've seen both sides of it. I'm going to stay out of the cellular space other than to say, you know, I've seen both sides of it. I give him credit in that I see a lot of people state opinions without a lot of backup for it. While I may not agree with all his assumptions, it is a very thoughtful report. The main thing I would say about it is my kind of high level assumption was reading it, it sounded like it based it off, we have to do it like pharmaceutical or like mammalian cells have been grown to date. And if we do that, the economics don't work. I don't disagree with that. The companies that are succeeding, you have to do it in a system that looks more like a microbial fermentation. What we're building, you know, do I think we can do that today? From what I know, no. You know, I think we can make that, but even in the things I've seen in the space, a couple of years ago, people would say there's huge, you know, we'll never get there on the media components. And it was because we were using pharmaceutical grade media made in small batches. I think as you've looked, most of the things I've seen indicate, you know, most of these media components are a lot like the proteins and other things we can make at large scale. So I think that's one area that's come down.
Mark, what about all the various custom steps in DSP that are possible? The question that came in from an anonymous user was, what drives costs up for users? And customization obviously is one of those things. Yep. Can you talk about other things that might drive costs up? And what do you plan to do for DSP? Do you plan to allow enough open space around you that bulk users might be able to come in and co-locate some of that DSP?
You know, potentially. So again, let me talk about our DSP. From the time you come out of what I like to tell investors is a modern industrial biotech DSP for at least what we're trying to make, proteins and things like proteins. It's a high-tech version of sorting a pile of rocks. What you want is the middle-sized rock. The organism is a boulder in the terms of microbiology, and the salts are pebbles. You want mid-sized rocks. So you need to remove the organism as efficiently as you can, assuming it's a secreted protein. Then you need to hold your product, your protein or other thing, while you wash the salts and stuff out. That can be done with centrifuges. We believe it's done more efficiently. And again, when I keep talking efficiency, I'm not saying, can I make it? What's my yield? When I'm running a facility for a year, I spend a lot of money to ferment a protein. The difference between recovering 65% of it and recovering 85% of it is huge. It's, in simple math, about 15, 20% of the cost of the product. So we're set up for high throughput, high efficiency recovery. So because of that, we're encouraging people away from specialization unless they need it. Are you doing it because it's what you're comfortable with, or are you doing it because it's what's needed? We think the most efficient network, just like us, to some degree, we're public. Our approach to manufacturing is somewhat of a hedging strategy. We're not betting on one company. We're betting, I would argue, on a group of about 30 we believe we can host and think that are in the right size range or different that you know, again, if you look at even the pharma industry, are most of the successful CMOs really only tied to one product for one company? Or if they built a network that can feed, you know, and it's hard. How do I get the economics of feeding the most or serving the most of them while keeping my costs somewhat constrained?
I think some of the recently, I'm going to use a terrible pun here, Mark, so I hope you'll forgive me. Some of the recently grown CMOs have designated centers of excellence for different types of bioprocess, right? So they don't try to be all things to all people at every location. And I think that's an interesting development.
Yeah, I think that's fine, because I think a lot of the ones you're talking about are more the scale up side than the actual manufacturing. And it's that knowledge base of expertise that I think you see specialized more the center of excellence concept on the development side than you do the manufacturing side.
I have one last question for you, Mark, and really want to thank you for spending this time with us today. It's very much appreciated and enlightening, I know, for all of our listeners. We got asked this question at the American Biomanufacturing Summit in California a couple of months ago, which was when you need a biomanufacturing technician workforce and you know you've got about 2 years of construction time, what's the right time to develop those kind of workforce partnerships to make sure that that team will be there when you're ready to go?
Well, I can tell you what we're doing, and it's a roadmap that I've used at other facilities. So planning, we're talking now. Facility is not scheduled to start up for another 18 months, and we've already had detailed discussions with our local trade organizations of how are we going to source people? Can they help us with curriculum, the universities and things? We're working with them to set up a program that as we bring people in, that we're going to have different levels. What I'm used to is 2 levels of training, usually a month to 2 months for pretty much all employees. The question is, do they need basic operator training? Do you know when a pump's cavitating? Do you know how to fix a control valve? Do you basic kind of operator, one of the things that a 5+ year operator, even in a wastewater treatment plant or a pulp mill or some process industry would come in with. Some groups have that, some don't. So we want to either do refresher or other things to make sure we get everybody up to a certain level. And then, probably a month or so of very specific training around our equipment. What is aseptic fermentation? More specific, health and safety items. What's important in our quality? How do you run? Because you want everybody, even if they turn out being a fermentation operator, you want them understanding the whole plant and why we run the spray dryer the way we do. So we're envisioning probably a couple months of training during that. And we'll probably hire most people probably at least in the range of 3 to 6 months out. We'll start hiring plant management supervisors before that. This is still being finalized, but usually you don't want to hire everybody at once because you'll start bringing the facility up slowly in pieces and making sure everything's works. So, but that's definitely a template I've used before. I guess the last thing I would say, and this is a push for more biomanufacturing in general in the US, you can teach this stuff and we need to, but unless you have a place for them to run it and have a job, it's all going to be lost. And that's not just for us, that's for others. The best training, I'm going to tell you myself personally, I've gone to a lot of training. Well, foreign language is a good example. Other than Portuguese, where I got thrown into Brazil for a couple of years, where I'm barely functional, I've learned a lot of things, but if you don't use it, you'll lose it in a year or two. So the training programs need to be layered with a continued push to build this manufacturing so people want to go in and do that trade.
Makes a huge difference when students have seen a bioreactor before they come to work the first time. Mark Warner, thank you so much. Our friend Mark Warner is co-founder and CEO of Liberation Labs with some very exciting developments. Mark, thank you again for your time.
Appreciate it. It was great. Thank you.
Talk soon.